The overall goal of this procedure is to establish a novel method for drug efficacy testing with surgical specimens of brain tumours, termed the tumor explant method. This is accomplished by first securing freshly removed surgical specimens of malignant gliomas during surgery for preclinical experiments. Next, the samples are transported to the Tissue Procurement Core facility to separate them for clinical diagnoses and research purposes. Then, in the research laboratory, tumour explants are dissected into multiple smaller tissues and transferred into optimized medium. Finally, intratumoral injections with drug candidates are performed and the explants are incubated for 16 hours. Ultimately, results can be obtained that reveal the efficacy of treatment through hematoxylin and eosin staining, as well as immunohistochemistry with cell type specific markers. We can also evaluate drug efficacy in vitro using flow cytometry and neurosphere forming assays.
The majority of the current drug efficacy assays leave a gap between the drug efficacy in the current preclinical experimental models and the efficacy in patients. In particular, when we test drug candidates on solid cancers such as malignant gliomas, we cannot underestimate intercellular as well as intracellular signaling exchanges. In order to avoid breaking microenvironment for solid tumors, we established an organotypic culture method with our tumor explants.
After tumors are removed from the patients, the samples are transferred from OR to Department of Pathology.
We divide the tumor chunks into two pieces. Tumor sample from one piece is used for frozen sectioning and paraffin embedding for clinical diagnosis, and the rest of the tumor tissue is sent to the research laboratories to be used by the researchers.
Using the method, we can preserve the microenvironment of the tumor with adjacent tumor, endothelial and immune cells, as well as normal brain cells that are either surrounding the tumor or migrating into tumor cavities.
Prior to processing glioblastoma tissue, prepare liquid medium and agar solution. Mix them in a 1:1 ratio. Then add 1 milliliter to each well of 6 well plates. After receiving fresh glioblastoma multiform tissue, transfer it to a petri dish using forceps and carefully wash it 3 times with 5 milliliters of ice-cold 1x PBS. With a surgical blade and forceps, cut serial sections around 10 mm in diameter of the specimens from the dissected samples to create tumor blocks. Transfer each block into one well of the 6-well plate containing agar medium, allowing it to have enough exposure to air to keep it viable. Next, using a 1 milliliter insulin syringe and a 28 gauge half-inch needle, inject tumor blocks 3 times with 5% DMSO or with 2.5 nanomolar temozolomide or a novel anti-cancer drug candidate, and incubate for 16 hours at 37 degrees Celsius in humidified air containing 5% carbon dioxide. After the incubation, wash the blocks 3 times with 5 milliliters of 1x PBS. Then, in 50 milliliter tubes, fix the blocks with 10 milliliters of 10% volume per volume formalin for 24 hours. Paraffin embed the tissue and cut 4 micron sections. To deparaffinize and rehydrate the sections, place the slides in a rack and incubate them in the following solutions: xylene 3 times for 5 minutes each, 100% ethanol 3 times for 5 minutes each, 95% ethanol once for 5 minutes, and 70% ethanol once for 5 minutes. Finally, rinse in running tap water for 3 minutes. Please see the accompanying written protocol for details on antigen retrieval and inhibition of internal peroxide. To block the sections, cover the tissues with 10% normal goat serum. Transfer the slides into a humid box and incubate for 1 hour at room temperature. Wash the slides 3 times in 1 times PBS for 5 minutes each. Next, add primary antibodies such as human-specific caspase 3 or Ki67, diluted with 1 times PBS. Then, incubate overnight at 4 degrees Celsius. The following day, rinse the slides in 1 times PBS 3 times for 10 minutes each. Next, cover the tissues with 2 to 3 drops of HRP-labeled secondary antibody. Put the slides in a humid box and incubate for 1 hour at room temperature. Wash with 1 times PBS 3 times for 5 minutes each. Develop the samples using a Chromogen DAB kit following the manufacturer's instructions. To counterstain, immerse the slides in hematoxylin for 10 to 15 seconds. Then, rinse with running tap water for 3 minutes. Dehydrate the samples by immersing the slides in the following solutions. Add a mounting reagent and a cover slip, then use a fluorescence microscope to image the slides. The T1-weighted MRI image with gadolinium enhancement in this figure demonstrates an enhanced glioblastoma multiforme or GBM tumour in the right frontal lobe of a patient before surgery. The post-operative image confirmed subtotal removal of the lesion after surgery. Hematoxylin and eosin staining demonstrates the presence of necrosis, pseudopalisading cells, and microvascular proliferation. Thus, these tumours were histopathologically diagnosed as GBM. To investigate if these tumour explant samples could be reliably utilised for the purpose of a drug efficacy test, we tested the effect of the drug temozolomide, or TMZ. Immunohistochemistry of TMZ-treated GBM tissues demonstrates a substantial reduction of Ki67 positive tumour cells in comparison to the control samples. In turn, immunohistochemistry with an apoptosis marker, caspase 3, did not yield any significant difference between TMZ-treated glioma specimens and the control samples. Treatment effect with temozolomide can be further characterized by combining this explant assay together with either in vitro culture or flow cytometry. We can compare this explant method with xenograft transplantation and in vitro sphere-forming assay for screening of novel anti-cancer agents.
Once you obtain the tumor tissues in the laboratory, the procedure for this experiment can be completed within 1 hour. This new assay is relatively easy to perform and reproducible. After dissociation of treated tumor explants, we can combine with in vitro experiments.
A major advantage of this assay is that it preserves the tumor microenvironment. We obtained positive data with temodar-treated tumor explants, which were consistent with the outcome of the same patients who underwent temodar treatment following surgeries. Lastly, this assay is applicable to other solid cancers and therefore may have the potential to accelerate identifying novel chemotherapeutic agents for solid cancer treatment.